Welcome to section 3.4, the cell membrane. In this section, we'll explore one of the most vital structures in all living cells, the plasma membrane. It's far more than a barrier. It's a dynamic gatekeeper and communicator between the inside of the cell and the outside world. By the end of this section, you should be able to explain the fluid mosaic model and describe the key roles that phospholipids, proteins, cholesterol, and carbohydrates play in membrane structure and function. The plasma membrane is a selectively permeable boundary composed of a phospholipid bilayer with embedded proteins, cholesterol, and carbohydrates. Its roles include protecting the cell, allowing communication with the environment, offering structural support, and regulating what enters and exits the cell. It's a vital structure that keeps the internal environment of the cell stable, what we call homeostasis. The fluid mosaic model, introduced in 1972 by Singer and Nicholson, provides a framework for understanding the cell membrane. Picture a mosaic, many small pieces forming a complete picture. Now imagine those pieces floating in a fluid. That's your membrane, phospholipids and proteins drifting laterally within the bilayer. This flexibility supports the membrane's ability to self-heal, adapt, and maintain selective permeability. Phospholipids form the foundation of the membrane. Each has a hydrophilic head and two hydrophobic tails. In water, they arrange themselves into a bilayer, with heads facing out and tails tucked inside. This arrangement creates a barrier that separates the cell's interior from its environment while still allowing essential substances to pass through. Cholesterol is interspersed among the phospholipids. It plays a key role in moderating membrane fluidity. At low temperatures, it prevents the membrane from becoming too rigid. At high temperatures, it prevents it from becoming too fluid. This helps the membrane remain stable and functional under a variety of conditions. Proteins embedded in or attached to the membrane perform many essential functions. Integral proteins span the membrane and include channels, pumps, and receptors that allow communication and material transport. Peripheral proteins found on the surface assist with cell signaling and maintaining the cell's shape. Carbohydrates are found on the exterior of the plasma membrane, attached to proteins or lipids as glycoproteins and glycolipids. These carbohydrate chains are involved in cell recognition and adhesion, crucial for immune response and tissue formation. Think of them as the ID tags of the cell, helping distinguish self from non-self. Viruses have evolved to exploit membrane structures to infect host cells. For example, the flu virus uses a protein called hemagglutinin to attach to glycoproteins on the surface of respiratory cells. This is why certain viruses infect specific tissues or species. By understanding these interactions, scientists can better design vaccines and antiviral therapies. The plasma membrane isn't just a protective wall, it's a responsive interface. It supports communication, signaling, structural integrity, and selective transport. Thanks to its flexible, fluid, and mosaic like composition, it adapts to the cell's needs in real time.